tRNA works as the cell's code breaker. Not that it has a brain and is really good at cracking codes, it just so happens that it has two special ends to it. Each one of these structures is a molecule of tRNA, and it's made of RNA nucleotides. We don't care about most of them, but at one end it has a sequence of 3 nucleotides, and at the other end it has a particular amino acid. Each tRNA that has UAC is going to have the same kind of amino acid. Each tRNA that has GUA is going to have the same kind of amino acid. Now, each of these 3-letter sequences is known as an anticodon, not because it's against anything, but because it matches or is complementary to these 3 nucleotides on the mRNA, which are known as a codon. Whenever you see the ON at the end, that means unit. So codon is the coding unit. This is what tells us which amino acid we need to have next. Now, how do we break the code? The tRNA has the complementary sequence and the correct amino acid. The tRNA has no idea what's going on here. It's not directing anything. It just so happens that it matches the codon on the mRNA and brings an amino acid along with it. So where we have AUG on the mRNA, this codon, the tRNA that has the complementary sequence UAC will bind to that and will bring along with it this particular amino acid, which happens to be methionine. The next codon has CAT. Therefore, the tRNA that has the matching anticodon will be GUA, and it will have a different amino acid. This one is GCA, so the tRNA will have CGU. This one's GCU, so the tRNA anticodon will be CGA. Now, if you'll notice, I put the same amino acid on both of these tRNAs. This is because we have 64 codons, 64 combinations of nucleotides, but only 20 amino acids. So some codons are going to give rise to the same amino acid. There's going to be some duplication. And that's a good thing, because if we make a mistake when we're making this mRNA and we mean to put GCA, but we accidentally put GCU, not that we do, but the RNA polymerase, then even though there's been a mistake in the transcription, the amino acid will be the same and there will be no change in the resulting protein.